Welcome to this presentation on brewing microbiology given to the Brewers Congress meeting on the 28th of November 2018. Now, our title for this presentation is rather sort of unfocused, so I've tried to give that a little bit of a more modern and also comparative focus. So really to say what we're looking at in brewing microbiology is of course the future, but with a degree of relationship to the past and the lessons that we've learned. Now, as a quick summary of our methods, let's just quickly run through some of the different techniques which you may well be familiar with, starting with microscopy, which we can see here an example of a yeast flocculating, a lot of characteristics we can see, and we can determine whether we've got contamination in the background, any deterioration of the cells, a whole variety of information that we can quickly see and instantly take a judgment on. But of course, microscopy isn't the only approach that we can take, and sometimes you want to see what's there in a growing condition, what's there at low levels and so on. So we might well use plate cultures. Now here's a plate culture which we wouldn't want to see anywhere in a microbiology, but in fact it's from an air settle plate. So it's everything there that would be in the atmosphere. Plates are useful, but they do take a time in order to grow. It could be 5 days, maybe even 7 days, depending on the organisms. So to some degree what we're thinking is, are there more rapid analysis techniques that we can use? Luminometry is one where we might be able to take a swab and put the swab into the luminometry detector and give us an indication of what level of microorganisms are present. And this would be a reasonably rapid technique. It would allow us to give a fairly quick feedback to the processing. What it can't do is tell us the absolute levels. It will be comparative. If it's high, if it's low, whether we need to clean again, and so on. One of the techniques that we can use in analysis of, say, yeasts, particularly in developing and applying yeasts and choosing yeasts and so on, is physiology. So in this particular example here on the left, we've got yeast growing and showing a growth curve in conditions of a 1040 wort. On the right, we've got conditions of a 1080 wort, and we can compare the kinetics of these and judge whether a particular yeast is suitable for a particular application, whether it would be high gravity or it would be a particular growth medium. Comparative to that, we would have broth and forcing tests, where we might put our sample of yeast or take a sample of our brew and force it to see whether there were organisms growing there, what they were, and the characteristics of them. But what I'd like to focus on a bit more in this presentation is the molecular biology techniques, whereby we can look at some of the DNA profiles, some of the DNA sequences that we can extract from a microorganism. And we can get the standard DNA sequences of A, T, G, C and so on from a sample of DNA that we've purified and perhaps amplified. Now, some of the applications of those techniques are really varied, quite varied. For a lot of applications, it might well be to identify an organism. It might be to identify a bacteria using a 16S ribosomal gene, or a yeast using a 26S ribosomal gene sequence. And we can compare the sequence that we've isolated and purified against a database sequence and use that to identify and say, well, we've got a lactic acid bacteria here, we've got a pediococcus, and we've got a particular yeast, it might be Brettanomyces, or we might want to check the identity of our own brewing yeasts. So there's a whole variety of value that we can get out of using molecular biology techniques. We can look at, in other procedures, at sequencing the whole genome. So these ribosome genes help us identify a particular organism, but if we sequence the whole genome of the organism, then what we can look at is all of the genes. And we can look at specific genes, and we can see whether it's got a POF gene that's active or not. We can see whether it's got a maltodextrinase uptake gene which is active, and so on. So that allows us to get a much wider view of particular organisms that we've got present. Now, I just mentioned that there are other techniques which are very rapid as well. So MALDI-TOF is a technique used in a lot of clinical applications where people are looking for diagnosis for disease. There's a big database of the signatures that come out of this MALDI-TOF, and it's very quick to use, within minutes really. But it's not quite fully developed for food use or brewing use yet, but I suspect that we will see that being applied in the not too distant future. Now, a couple of applications that we would have in molecular biology for brewing would be to identify and apply to a particular gene that would be of concern. So the lactic acid bacteria are often a danger if they have hop tolerance. And there are certain species that have that, but there is a worry that there is transfer of these genes between different bacteria. So the HORA gene is one that is signature for hop tolerant product, and it will allow those bacteria to grow in wort with high levels of hops. So if we can target that gene, we can say whether or not a sample that we've got has a hazard. And in this particular demonstration here, we can see that we've got some positive controls of lactic acid bacteria, lactobacillus, and the cycling here is the progressive amplification of the DNA. And a positive result would show an increase, as these lactic acid or lactobacillus samples do. A negative control would show no real increase at all. Now we've got 3 samples there of a mega blend, which is a mixture of wild isolates of yeast and bacteria mixed together. And in these 1, 2, 3s, we've got different conditions of growth periods. And they are all showing an increase in the presence of this gene. So it shows that those genes would be present. It shows that those bacteria would be likely to grow and produce their relevant products. In likely chance would be lactic acid bacteria, or lactic acid, which would cause flavour characteristics. Other applications might be for phenolic off flavours or diastaticas, a very contemporary concern at the moment. So if we can choose the genes that are relevant to those and monitor them, then we can identify whether they're present in our particular sample. Now, when we're looking at identifying these particular things, that's a relevance for day-to-day processing, but we might well be looking to identify isolates from the wild or from some other location. So in this case, here's an example of taking a fruit from the wild, and in this case it's overwintered, so we've got colonies in a bit of a shrunken fruit that we can isolate. We can put them onto agar plates and culture them up and get colonies, and we can identify the consistency of those colonies or the characteristics of them, and we can further do some microscopy on it. So it allows us to say, oh well, okay, here's a yeast from the wild, here's a yeast that we might be interested in. Can we then go further and identify that? And in this particular case we found 5 different yeasts from that particular sample. Here's the magnified sample, there's the surface of the fruit, and here's the colonies. Interestingly, quite filamentous, although there will be standard yeast growth in there, but don't forget, yeast and filamentous fungi often interchange as a dimorphic condition. So we isolated 5 different yeast from this, including 2 Saccharomyces cerevisiae strains or species. Now we then further processed those to look at the physiology. How would they react in fermentation conditions? And we found that they produced different levels of alcohol, ethanol. A control yeast gave 4%, yeast number 1 gave 3.5%. So that might well be worth looking at in maybe a pure culture condition. But in many cases we might want to mix in some of these yeasts. So we might say that number 1 and maybe number 3 with a good flocculation ability would be a good combination. But these processes allow us to then take further steps and not just have to rely on a mixed unknown culture. We can identify and further do the physiology to select what we might be interested in. Another application we can do is see the consistency of a yeast that we've been using for over a period of time. We might be repitching it a number of times, and we want to see whether there's any genetic change in that. So we can do an isolation from a yeast at a particular time and compare it with an isolation of a week ago, a month ago, a year ago, and see how consistent that is. So in this particular case we've got 97% consistency, which we might say is very good, but of course if a mutation has occurred in a critical point of that sequence, it could be a big change. But we might be looking for, say, flocculation genes. So in this particular case we're looking for the comparison between different isolates of flocculation genes, and flocculation will allow clarity, and therefore if that does change we might have a problem. So this would be part of perhaps a routine longer term processing rather than every brew or if we've got a problem. Now other workers have done much more extensive work and studies on a whole range of yeasts. So in this particular group they've taken over 100 yeasts from different sources, wine, beer, overseas sources and so on, Belgium, Germany, US, mixed cultures and such. And they've done the sequencing of compared them, and what it's brought out is that there are common groupings. So wine groups very differently from beer. Beer actually has two big groups of different characteristics. And within the beer group, we've got groups from Germany, Belgium, which are very similar. The US groupings, yeast there, similar but different than the Belgium-Germany one and different from the Britain one. This group has also done a comparison of the physiology. So here we've got ethanol production. So it's comparing those different groups for their ability to produce ethanol, and as we might expect, the beer group is able to produce maybe up to 10-11%, whereas the wine group or the mixed group or the spirits group from Asia can produce higher levels. But when we look at maltotriose uptake or fermentation, where we'd expect that to be high, yes, we do have it high in the beer group, but very low in the wine group. So sort of conforming to expectation, but these are the physiology studies which will compare and correlate to the genetic studies. Now another group has done a similar selection of many yeasts, grouped them together again and found a similar sort of split between Irish and English ales and German and Alt Kolsch beers. Also a separate split of wheat beers. So we've got different characteristics between these groupings. There are other groups, of course, sake and wine and so on. But when we look more closely at the beer groups, then what we find is not just the separation of the groups, but the genetics of particular features and the physiologies that we might be interested in. So the POF off flavour, the phenolic off flavour, is a feature that we don't really want in most standard beers, but we do want in a wheat beer. And when these people have looked at this and found that the wheat beer group has very active POF genes, but in the English ales and the German ales, those particular genes have mutations in, so they are not active, and therefore they don't produce that POF flavour. And that would have been selected over many decades, centuries probably, in order to give the flavours that we associate with those beer styles. Maltotriose uptake is also distinctly different for the wheat beer groups and the standard groups for our standard ales. So there are beginning to be a body of knowledge into the compositions of yeast groups, into the genetics of specific genes, or features of specific genes that give characteristics to particular styles. Now here's another application of genetics where what we've done here is go back to that mega blend, so a mixture of yeasts and bacteria from environmental sources. What we were interested to know was, well, a couple of things. One was whether there's any specific hazards in that, any food poisoning bacteria, which could cause a problem, but also what the distribution of species were. So we use next generation sequencing, which is a procedure that allows us to identify the range of particular ribosomal gene sequences in these samples. And what we find in the Mega 1, 2, 3 of the progressive fermentation is that we've got initially a predominance of acetobacters, fewer lactics initially, but the lactic acid bacteria develop as the growth pursues, and there's a bit of a balance at the end. So we've got quite a range of acetics, a good range of lactics and pediococcuses, with one maybe more unusual Pantania. But what we didn't find were any food pathogens, which is obviously a positive result. We did the same with the yeast from this group, so we looked at the presence of different yeast. We've got Brettanomyces here initially in the groupings, less so Saccharomyces, a couple of pitchier samples, but towards the end of the fermentation we do get Saccharomyces being fairly dominant in the system, as we might expect. Another application here, we did some research into the possibility of using spent grain from brewing in bioremediation. So can we use spent grain to enhance the digestion of hydrocarbons in, for example, oil spillages? And we looked at different samples at different times. So we've got 0 time, we've got 5 days, 12 days, 21 days. And what we're considering here is what species of bacteria are dominant and are they changed by the presence and inclusion of spent grains. And we did find, yes, there are particular bacteria which are enhanced in those conditions. So particularly Pseudomonas putida which is also well known as being able to digest hydrocarbons. So if we can identify these organisms and maybe purify them, we can use them and make recommendations for other uses of spent grain. And we did the same again with the fungi in that group, and we found particular fungi species which were particularly enhanced by the spent grain, in this particular case Pizia species. So our options of using microorganisms in the future have expanded, and there's a variety of things we can do and we want to support with techniques. So we could use pure yeast. Obviously we do that already, but we could go back and look at what people did many years ago and use mixed yeast. Tended in the last decades really to purify our yeast and select one out of a mixture, but maybe mixtures have advantages, providing a variety of characteristics and abilities. So I think there will be opportunities to go back to using that and looking at historical sources. We could add bacteria in. People are doing this in sour beers certainly, but we need to know what bacteria we're using. We don't want to be using something which is potentially hazardous. So there's a bit of need for the right stock, the pure stocks, or the identification to be included in that. And bearing in mind that mixed microbes often have a sequential development, so we have a community develops which succeeds progressively. We've got, say, in an open fermentation, Hansennia spora might grow first, not being terribly resistant to alcohol, but able to grow on the high sugar levels, Saccharomyces taking over, producing alcohol, reducing the Hansennia spora group. But then when the Saccharomyces has finished fermenting the fermentable sugars, Brettanomyces coming through and maybe processing some ethanol into alcohol or other residual sugars. So this sequence could be managed, it could be used to produce particular beers or products with different characteristics at different times. We could just do a wild fermentation, no microbes at all, and let the environment take over, but there are hazards to that and we need to keep a careful control over it if we're using it for a commercial product. Now it's interesting, going back to our very beginning and saying that there are possibly lessons from history that we could bring into all this. And here are some images, some early photo micrographs from brewers or brewing yeast, indicating a couple of features. One is that, well, some of these yeasts appear to be pure and some of them appear not to be pure. Some of them appear to have both a yeast and a hyphal characteristic. So there are probably more wild type characteristics in these yeasts, but some of them are pure. But then when we look in the background, we can see clear evidence of bacteria growing in collaboration with the yeast in a number of these. So it rather suggests that our historic beers, which were in many cases well accepted, may have had characteristics of various sorts from combination communities. So a few lessons now for the future in the sense of what are people doing. Well, there's a whole wealth of literature which is indicating the studies that people are undertaking and reporting, partly which relate to characterization of yeasts, but also modification of yeasts. So in this particular case, using microparticles and so on to modify yeasts, but identifying inter and intra-species and hybridization, which is developing yeasts to have different characteristics, combined characteristics, may be suitable for particular growth media and so on. Going further, we've got enhancement of particular flavours, we've got recombinant Brettanomyces here, we've got gene expressions being investigated, in this particular case for wheat beers, and in some cases mutagenesis. All of these will be developing yeast strains which may well become commercially available. It depends which part of the world we're looking at, but they are being reported and there's probably plenty of commercial operations undertaking many more. Now these are the studies which have been cited in this report briefly earlier, and I think just to finish on the future, to be able to say what are the options that we've got, how can we extend our existing skills, techniques and knowledge. We can generate a wider awareness of brewing microbiology, of course, wider awareness of the range of yeast strains and species that we can use, we can mix, we can incorporate into our system. Particularly, we can develop our knowledge and understanding of yeast genetics and how we can control yeast genes. It might be media, it might be temperature control, it might be a whole variety of new materials that we can incorporate. If we can use dual and multiple strains, then we can broaden the potential characteristics of our products. And lastly, if we can use modified, possibly engineered, but maybe hybridized strains and so on, we can have a wider range of activities again. So all of these will develop in the future, I'm sure, and maybe not so many years to come. It's a matter of looking out for them, becoming involved in them, or at least the knowledge. And if we don't do it, other people will. So the brewing community will expand, will take on these technologies and apply them. It's something to look forward to, something to be aware of, and something to be conscious of the both not so much risks as its applications and how we can use those positively.